Bueno, esa ley que empezó a tratar a mi bueno, buenas tardes. Comenzamos el ciclo de seminarios 2023, no es original, y hoy tenemos el placer de tener al profesor Dr Desaulniers, que bueno, pudimos darnos una charla acá. El Dr Desaulniers, profesor en Ontario, Canadá, y bueno, hizo su carrera en Ontario, hizo su doctorado bajo la supervisión de Tim Chao en un tema de nucleótidos, y luego estuvo una tarea prolongada en la Universidad de Michigan bajo la supervisión de la doctora bueno, temas distintos de medicina. Y hoy nos va a hablar de la síntesis de Azobenzene, su control nucleotide. Bueno,
Okay.
breve pero conciso.
All right. Thank you. Yeah, thank you very much. Thank you, Professor Juan Rodriguez, for that introduction. I don't think I understood most of it, but I think he was saying good things.
Good things.
Good things? Okay. All right. So it's a real pleasure to be here in Buenos Aires and to be able to present the work I do at Ontario Tech University, where I work with oligonucleotides, so molecules like RNA and DNA. And specifically, I'm going to talk to you about these molecules that we use in our lab to control the function of oligonucleotides. So throughout this talk, you'll see the ability of us to use something like azobenzene, its ability to isomerize between cis and trans, and then how we create different derivatives and use different colored lights to control the oligonucleotide function. So I'll be talking about that throughout this talk. But before I do that, I want to give you a bit of background about my university, who I am, and also about oligonucleotides, since I understand that perhaps some of you don't know everything about oligonucleotide therapies in the field right now. Okay, so just a little bit about, so as Professor Rodriguez said, I'm from Canada. So actually, I live very close to Toronto. So Ontario Tech is actually very close to Toronto, about 50 kilometers away. We're a very vast country. You've probably heard of Vancouver before. We had the Olympics. But as you go from west to east, things can change very, very drastically in terms of the landscape. So in this area on the western side of Canada, if you ever have a chance to visit, we have some beautiful mountains, some beautiful scenery. Then as you head more towards the east, we hit Ontario, which is our most populated province. And I know, of course, football is the #1 sport here, but if some of you like basketball, basketball is very important. And we won the NBA championships in 2019. And of course, hockey is probably our #1 sport in Canada. And as you go east, this province here, Quebec, is a French-speaking province. So if you have a chance to visit Canada, please do. So I was born and raised in Canada, and I went to Western University and I got my bachelor's degree. And here I was very interested in oligonucleotides as an undergraduate. So I went and did my PhD at Wayne State. During my time there, I learned how to work with RNA, how to synthesize RNA. And one common modification that I was working on was something called pseudouridine. I won't talk much about it today, but pseudouridine is a natural carbon-carbon glycoside, and it's actually used in some of the mRNA vaccines that many of you have taken. It's more stable, and it actually evades the immune system. So anyways, I was looking at studying RNA structure and function using things like NMR. Then I did a postdoc with NMAP, where I focused on small molecules, synthesis of small molecules as protein-protein inhibitors. And I learned also a lot of biology during my postdoc. And then I started my independent career at Ontario Tech, where I've been designing chemically modified oligonucleotides for various applications. Ontario Tech University is actually one of Canada's youngest universities. So, this university, of course, has a great history. Well, right now, we're celebrating our 20th year. So we were founded in 2003. We have around 10,000 students. And as I mentioned, we are, not so far from Toronto, Canada. Okay, so before I talk about some of the chemistry that we're working on, I want to show this slide, which is a slide that highlights the ability of us to target various diseases. So this is known as the central dogma of biology. So DNA is transcribed to messenger RNA, and then messenger RNA is translated to various proteins. So in a lot of medicinal chemistry, small molecules are in the market, and often those small molecules will target various enzymes or proteins and inhibit these protein-protein interactions. And that's very good, but it turns out that not all proteins can be targeted with small molecules. So another approach involves targeting the messenger RNA before it's translated so that we do not get that protein made. And because we know the sequence of all the mRNA transcripts, we could, in theory, design, unique transcripts to target individual messenger RNAs. So it sounds very simple, but it's a little more complicated than that. So one way to do this involves something called the RNA interference pathway. So this was a pathway that was discovered about 20 years ago. And it's a pathway that all of our cells use, plants use, higher ordered organisms use. And essentially what happens is that there's natural double-stranded RNAs inside our cells. It's cleaved by an enzyme called dicer. And then now we have these short interfering RNAs that are loaded into this enzyme complex called RISC, which stands for RNA-induced silencing complex. The two strands, as you can see here, green and blue, the passenger strand is removed. And now we're left with a guide strand that's now attached to the RISC. So this guide strand can now bind to that messenger RNA. And now you can get cleaved messenger RNA, and then you get gene silencing, and you don't get that protein made. So it's a very effective way to reduce or to eliminate that protein of interest. And it turns out a lot of diseases are characterized by high levels of specific proteins. So we could potentially use this pathway to target specific messenger RNAs. So this was discovered, a little over than 20 years ago. And subsequently, the Nobel Prize for this was given in 2006 to Fire-Mello. And a lot of companies and clinical trials were on the market in order to target various diseases. But unfortunately, around 2010, 2011, a lot of these molecules failed, and a lot of money and resources had gone into them. And then there was loss of confidence in the field, and only a few companies remained. Fortunately, some of the big companies like Alnylam decided to keep moving forward, even though they didn't have a product. And then the first product was expected and was actually approved in 2018. So it took almost 20 years to develop this product. And currently, there are actually 5 US FDA-approved siRNAs. I'm just showing you 4 here, and they target rare diseases. And the future looks bright currently because now they're looking at targeting more common diseases like cancers and metabolic diseases. And they're learning some of the lessons learned from the failures in order to push and move forward some of these therapeutics. So, you know, we've seen a lot of excitement with things like mRNA vaccines. Here's an example of an RNA therapeutic. So I think the future is bright for RNA type therapeutics. Just a little bit of word on how some of these function. This first drug that was approved in 2018 is called Onpattro, and it targets a protein called TTR, and in individuals, they have a mutation which causes this TTR protein to aggregate, and you get these amyloid deposits that can be very toxic to the patient. So this siRNA targets the mutated TTR, and then what happens is that you remove these amyloid deposits, and then you get fewer amyloid deposits in tissues, and then it resembles a healthy liver. And this is, you know, the sequence of the siRNA, and I'll talk a little bit more about it. But you know, it's a modified sequence where we have these 2' methyl groups, and basically what I'm saying is that all of these siRNAs moving forward have a high degree of chemical modification, and where the field is trying to figure out how to best modify them to elicit the best effect. RNAs have trouble crossing the cellular membrane, so these are encapsulated in something called a liposome, which helps it bring it across the cellular membrane. A more recent example includes something called Givlaari that was also approved just a couple of years ago, and this targets an enzyme called ALAS, and it leads to overexpression of a neurotoxic compound called delta aminolevulinic acid. So it's a metabolic pathway where there's too much of it, and they designed this siRNA that targets it so you can restore them to their proper levels. And this is actually very interesting because they've developed this molecule here, and I show this because I know there's some sugar chemists here in the room. This is a N-galactosamine derivative, and it binds very tightly when it's a trimer to this acetylago receptor, which is on the surface of liver cells, and you get very efficient endocytosis into the liver. Interestingly enough, if you have just one GalNAc, you get around millimolar KD binding affinity. If you have 2, you get micromolar binding affinity. So the magic bullet was 3 of them together through this linker, they can get a nanomolar binding affinity. So it's really quite remarkable. And then I'll show you some modifications later, but these are some unnatural phosphorothioates. And you can see here that they've 2' methyl them, and they've added some 2' fluoro. So in fact, this siRNA here is completely chemically modified. Okay, so in our group, in my group at Ontario Tech, we look at ways to modify the structure of RNA in order to overcome some issues like cell membrane permeability, some issues of stability. And, you know, so here's the structure of RNA. I haven't shown it yet, but RNA is this classical structure with, you know, these bases bound to these ribose sugars held together through phosphodiester bonds. So you can get actually nucleases coming here and attacking the phosphorus and then breaking the phosphorus oxygen bond. So RNA is a very labile molecule. So we've done some work where we've, you know, replaced the backbone with things like triazoles based on a peptide nucleic acid structure. That was some of our earlier work. Triazoles. This was nice because we could do the click chemistry. We've looked at amide bonds, which are much more stable compared to phosphodiester bonds, and these work well within siRNAs. More recently, some of our work has involved tagging RNAs with small molecules. So here's a molecule called cholesterol. So cholesterol is used to help move those sRNAs across the cell membrane. And we've also looked at, again, other cholesterol derivatives. We've also done some work at generating what are known as triester phosphates. So removing that negative charge and putting bulky hydrophobic groups, which again helps it get across the cell membrane. Now, how do we determine which groups to work on and how to modify? Well, it really came down to using these very simple spacer groups that I'll tell you about in a minute, and how we exploited that to generate these types of derivatives. Okay, so back to the siRNA picture where we had the duplex RNA that was incorporated by RISC, and then remember the passenger strand needs to get removed. So typically what happens is that an enzyme called Argonaute-2 will cut this passenger strand, removing it so that you now have a guide strand that can target the messenger RNA. What we had done is we wanted to thermally destabilize it. So we placed these very simple linkages and asked the question whether these duplexes could be removed simply by removing the hydrogen bond pattern at the middle. So very simple experiment. And we just added these polyethylene and these carbon-based linkers and asked the question whether they would still function. And it was sort of a drastic experiment because we're removing entire nucleic acid functionality from that center. So before I show you that data, I just want to show you some of the biology, what we do and how we analyze it. So in our lab, we grow cells. They're HeLa cell cancerous cells, and then we add a plasmid called luciferase. And so we add the plasmid, and it encodes this firefly protein, which lights up, and we can monitor this quite well. And then we can monitor gene efficiency or knockdown by designing RNAs that target luciferase. So if we have no siRNA, you'll see high levels of luciferase, but if the RNA is working well, then you'll see low levels. So we can gauge how well it works based on how low that bar is. And then we also do internal controls with other plasmids just to make sure that the cells are taking the same amount of plasmids per cell. So when we did this experiment here, what we had done is on the sense passenger strand, we did an experiment where we took this ethylene glycol spacer linkage and we replaced it within that middle region. And first, I'll just show you the wild type data. So this is just regular unmodified RNA. Here's our control, high levels of luciferase, and you can see here that we get low levels of luciferase activity, meaning it's working well, which is what is expected. When we add the spacer linkage, you can also see a very similar pattern, meaning that these are also working really well. A little bit surprising because it doesn't look anything like RNA. Nevertheless, we were happy with those results. When we treat it with this carbon linker, we lose some potency, but again, we can see a similar pattern, meaning that we get dose-dependent gene silencing. And then finally, when we try to replace 3 nucleotides with this very long linker, not as potent, but again, we see gene silencing. So you might think, okay, these are not very exciting molecules, and I certainly agree, but there seems to be what we call a high degree of promiscuity within this area. Maybe we can add small molecules or different functional groups. So what we've done further was ask a question whether we could place other groups with more interesting functionality like alkynes or alkenes within this area here to see whether we could retain function. And, just to make a long story short, if we have things like acetyl groups, allyl groups, alkynyl groups, or even things like aromatic groups, conformationally constrained groups like biphenyls, you can see the same pattern. We can see dose-dependent knockdown in all these cases, meaning that they're working quite well. So we were encouraged by this and said, well, maybe we can move away from biphenyl and put something a little more interesting. So here's our story of these linkages working really well. The biphenyl worked well, but then we wanted to ask whether we could place something called azobenzene and whether this would be an appropriate substrate within that region. And why we want to do that is to be able to potentially control the activity of the oligonucleotide function. And I'll get to that in just a moment. Okay, so I've told you already some of the issues with RNA-based tools and therapeutics. Stability is an issue. We talked about cellular uptake. There are other issues as well, like biodistribution. Where does it go in the cell? Immune activation and off-target effects is another dominant issue. You can have similar genes and you want your RNA to be very specific. But one thing that is not really talked too much about in the field is being able to control its activity inside the cell. And you can think also of maybe things like small molecules or any drugs in general. When it's inside the body, there's always a half-life associated with that. And sometimes with RNAs or other drugs, if the half-life is too long, or maybe you don't want that molecule to persist and be active all the time, it would be nice to have the ability to turn off the function of that molecule. So why this is important? So this is an important study that was done by Alnylam in terms of controlling RNA interference activity, and I'll walk you through this slowly. So Alnylam is the major company with the siRNAs on the market. They're based in Boston, Massachusetts, United States. And here's that GalNAc linker that I was telling you about. And here's the siRNA experiment. So they were targeting this TTR protein that I talked to you about very briefly at the beginning. When they inject the mouse with TTR siRNA, you can see that after 12, 14 days, you get very low levels of that protein, which is actually quite remarkable and unexpected. In fact, it can go on for many weeks and many months. But like I said, there has to be an ability to control and be able to reverse it, because sometimes you don't always want it to be active. So what they designed was something called Reversir. When they injected this at day 7, it competes for it competes for the seed region of the guide strand that was binding and displaces it from the messenger RNA. So you can see here, when they designed it, they could restore that TTR level back to normal levels. So this is sort of an artificial way to reverse siRNA activity, but it would be a little more practical to be able to reverse activity without having to administer another RNA or another drug. So this is where our work with azobenzene will come in. Now, before I do that, I just want to tell you a little bit about how we make RNAs in the laboratory, because maybe here not everyone is familiar with oligonucleotide synthesis. So there's actually some very beautiful chemistry that was developed in the 90s by Crothers. And essentially, we take these phosphoramidites and we can build oligonucleotides on a solid support column. And here's an example of a DMT phosphoramidite. Here's a dimethoxytrityl group, electrophilic phosphoramidite, and then we can build our growing oligonucleotide using this chemistry. It's quite robust, and here's how it works. So we can place our growing chain on starting from the 3' end on the solid support. It's usually like a control pore glass support. We detritylate with usually weak acid like trichloroacetic acid, and then we do our coupling reaction with our phosphoramidites. And we typically couple with things like, like thiolethyltetrazole or other similar reagents. And the coupling reaction really works quite well. Upwards of like 99% coupling. We now have a phosphate, and we need to oxidize that to a phosphate. But before we do that, we cap it, any unreacted groups here with acetic anhydride, so that when we continue the cycle, this will not further react, so we don't get mixed sequences. We oxidize the phosphate to a phosphate using iodine, water, and pyridine. It works really quite nicely. And then we detritylate. And then here we can add another phosphoramidite and continue the cycle over and over again. And when we're done the sequence, we simply need to remove this from the resin. And we can do that using ammonium hydroxide is typically used. And you can see the esters will get removed. Any protecting groups on the base will get removed. They're typically like benzoyl protecting groups. And then we can remove this group as well. So it's a really nice deprotection mechanism. And this is for DNA. With RNA, I don't have it here, but usually we use something like a TBS group, TBDMS group. And then after cleavage and deprotection, we have to do a sial deprotection, usually with something like HF. Now, the yields are really quite good. The coupling efficiencies are usually like 99%. Another nice thing about this chemistry is that when this DMT group is removed, I'll just go back quickly, you can see this beautiful orange color. That's the color of that trityl cation. So if your coupling is successful, you're going to install another one of these molecules. And then when this trityl gets released, you can monitor how much trityl is getting released and quantify that. So you can quantify the coupling efficiency. So it's really quite good. If something goes bad with the chemistry, you'll know because you'll get a loss of that deep orange red color or again. So that's oligonucleotide chemistry. So basically what we do in our lab is we custom make these phosphoramidites so we can place them within oligonucleotides. So a little bit about azobenzene now. So azobenzene is a unique molecule that can go isomerize between, trans and cis. So it's characterized by, a strong like pi to pi star transition in the UV area, and then a weaker N to pi star transition in the visible area. So in the case of azobenzene, we can isomerize it to cis using UV light, and then we can reverse this using visible light or heat to go back to trans. And azobenzene actually has been used in chemical biology for a number of applications, most notably things like peptides. Here's a nice example by Andrew Woolley where they had this sort of long elongated peptide. You can see the azobenzene in the transform, so it's inactive, but when they change it to a cis conformation with UV light, it can now come together and form this active alpha helix. So there's been a lot of utility in using azobenzene for this purpose. So in our case, because we've shown that we can place different molecules within that central region, we wanted to design a system where we could have azobenzene embedded within that guide strand. It would be active, but then using UV light, it would distort it in such a way to inactivate that complex's activity. So we could have an active RNA or an inactive RNA. And furthermore, we would have the potential to reverse it with visible light and go back and forth reversibly. So that was our hypothesis. Now, I don't have time to go through some of the other, we're not the first people to think of controlling RNA function with photolabile groups. There's been some really great work from other labs. But one common thing from all of them is that they developed these photo cages. So you've probably heard of photo cages, like typically like nitrophenyl groups that can be removed with like UV light, but those are typically irreversible. So you could only go in one direction. So using our system, we believe we can go back and forth both ways, which could offer some very useful, could be very useful. So how do we make some of these azobenzene phosphoramidites? So it's not so complicated. We can start with something like a nitrophenyl alcohol, and then we can reduce it with something like zinc under basic conditions to generate our azobenzene. And then really we have this diol. I mean, these are commercially available, and then we can simply add the DMT group on one end and then add the phosphoramidite on the other end. So when we started this, we had a couple different molecules where we had a single carbon benzylic species and then a couple different carbons between the alcohol. And then, we sort of did an evaluation of which ones work best. And then the one with the 2 carbons work best because, of course, they're more stable. But this is our DMT phosphoramidite that we can use for that oligonucleotide synthesis that I told you about. So how do we characterize some of these species? So we can use things like biophysical techniques. If we have a single strand, here's the RNA, it can flip to this different conformation. So we can monitor these things using like HPLC. So here's HPLC in the transform. When we add UV light, we can see shorter retention time, for example, in the cis form. And we can also characterize this by things like UV. You can see sort of a broad maximum around 340 nanometers in the transform, and then in the cis form, we don't have that. So we can monitor these using a variety of different techniques. So one of the first questions we wanted to ask was, can we activate these RNAs with visible light? once inside the cell. So I'll walk you through how we did this experiment. We can take an active siRNA, and then we inactivate it with UV light, and then we add it to the cell. So now we have an inactive siRNA, and then at some later time point, we want to add visible light to turn it on so that we can get RNA knockdown. So let's look at some of this data. So now so we tested 5 different siRNAs, and you can see the azobenzene here is all embedded within that guide strand. And rather than showing you all these different graphs and curves, we've done something here called a heat map. And it's really quite simple, where we have low luciferase activity. It's pronounced with brighter colors of yellow. If we have high luciferase activity, then we have blue color. So you can see with wild type, low luciferase, so it's working, and it really doesn't matter if we have UV visible or dark, which makes sense because there's no azobenzene. Now, let's just take a look at one of our siRNAs, like siRNA9. You can see when we inactivate it, you can see that we get a lot of blue colors, a lot of black colors, meaning that it's not working very well. We're getting poor gene silencing, but we can restore some of that activity when we add visible light. A lot of the blue is going to a lighter shade, or we're starting to see more yellow. And we can do this for 8 hours. So at 4 hours later, we can add the visible light and we can start to restore function. We can also do this for 24 hours. So you can see here a large amount of blue, meaning that it's inactive. And then after 24 hours, we add visible light, and you can see that we're getting this nice yellow pattern in general, suggesting that it's working. One thing though is that with this UV light, we had to cheat a little bit, okay? And I didn't tell you about that. We had to add UV light every 4 hours for about maybe 30 minutes, and that's not very practical. And the reason we had to add UV light every 4 hours was because the azobenzene and the cis form will naturally go back to the trans form after 4 hours. So it has a 4-hour half-life. So it's not that stable. And furthermore, adding UV light, we can already think of some problems. It's a little bit cytotoxic to the cells. Nobody wants to have UV light. So we wanted to move away from UV light. But before we do that, we first wanted to see whether this is reversible. So this was sort of our proof of concept. Did it work? Can we reverse it? So can we add our active RNA, add it to the cell, inactivate it with UV light, reactivate it with visible light, and then can we re-inactivate it and then reactivate it? So could we go through 2 rounds of cycling? That was one of the main important questions we wanted to ask. So we took one of the siRNAs and we did a 24-hour experiment. And of course, the UV light shows no knockdown, as you can see with the high bars here. Now, with the next experiment, we did UV, visible, UV. So we inactivated it, added visible light a few hours later, and then a few hours later added UV light and kept it inactive. And you can see in large part at the lower concentrations, we get no activity, some activity at the higher concentration. Now this next one, UV visible, was where we went through inactivation, activation, inactivation, activation, right? A little difficult to say, but you can see that at the end, we wanted it to have some activity. And that's certainly what we get with those gray bars. You get some dose-dependent knockdown. But if you compare that to the dark, where it was never inactivated before, you can see that it's not as potent, which makes sense, of course, because the dark control is going to be more active because it never became inactive. So we're very pleased with this reversibility. However, like I said, UV light is something we want to move away from. So we want to redshift, the pi to pi star transition that makes it go from trans to cis. So there's been some work in the literature showing that if you halogenate azobenzene with these chlorine groups, you can actually redshift it and not rely on UV light to isomerize. You can actually use colored lights like red or green light, which is much safer. So we actually adopted some methodology published by Dr Desaulniers, where they were doing a post-synthetic functionalization where they could take something like azobenzene and use like NCS and palladium acetate at high temperatures to tetrachlorinate this group. And I just want to say that typically to generate azobenzene derivatives, what typically has been done in the past is people will first functionalize it and then undergo that condensation. So this was a much different approach and more amenable to synthesis, reducing the number of steps, and it was quite efficient for us to use. And then really, we got 70% yield, then we could saponify the structure, and then like before, do the classic DMT protection, and then phosphoramidite, where we could now incorporate it within that oligonucleotide. So this really now is our new model where we have these tetrachlorinated groups, and now we can actually use red light to inactivate it, and then something like purple light to reactivate it. So we're not in the UV visible range. So I'll walk you through some of the data. We generated, again, a small library of this tetrachlorinated azobenzene within that central region like before. And I'll walk you through the data. So at 8 hours, if we add red light, we can inactivate it. And you can see here that similar pattern like before with UV light, no activity. If we add or dark control, so never had red light, so it's going to be on. Or if we remove the red light or add purple light, we can see activity. Okay, so we can turn it on simply by adding purple light or removing the red light. We can do this for 8 hours, and we can do this also for 24 hours as well. So up there it says 24 hours. You can't see it because this is in the way. But one of the problems though with this is, okay, it's good. We moved away from UV, but this tetrachlorinated group here in the cis form actually has a very short half-life. Azobenzene was 4 hours. This one here has a half-life of minutes. Okay, so these chlorinated groups, they're really quite big and bulky. They don't stabilize it well enough. So in order to keep it in the transform, we have to keep the red light on all the time. So in order for us to do the 8 hours and 24 hours in our cell culture room in the incubator, we had this red light on all the time, which was fine. It's not toxic, but maybe not so practical. But nevertheless, we could still observe this controlled effect by either having the red light on or removing it at specific time points. So we did this for, the different RNAs, and you can see, the very similar pattern suggesting that it's working quite well. And then this is our wild type control, again, showing no effect with the red light, which is exactly what we would expect. Okay, so the next thing we wanted to do was saying, well, we really want to increase the half-life of this tetrachlorinated azobenzene, but there's nothing we can really do about that. But we wanted to see whether we could use the same colored lights to get the same effect. So there's been some work done at doing these tetrafluorinated azobenzenes, and they go from trans to cis with green lights. And so there's been a few things in the literature. This is just a study done by FCEyN-UBA where they could control the antimicrobial activity by placing azobenzene. And you can see here with azobenzene, they use UV light. Here's the tetrachlorinated group, they use red light. Now this tetrafluorinated group is attractive because it actually has a very long half-life. It's actually been reported computationally to have a half-life of up to 2 years at room temperature. And certainly when we've done some experiments, we didn't do them for 2 years, but we observed in the monomer, the cis form, for very, very long periods of time. So we were attracted to this tetrafluorinated group. And some more recent examples, this is kind of interesting too. FCEyN-UBA and co-workers developed this tetrafluorinated group where they attached it to this. This is like an adenine derivative that targets like a casein kinase 1. It's involved in circadian rhythm. And so this could be very useful for targeting things like jet lag if you're on a long flight and traveling across multiple time zones. And essentially, you could take one of these derivatives and it could help restore your clock when you land, for example. So maybe in the future, we'll be taking some of these for long flights. Here's another example where they developed this matrix. I find this pretty interesting. And here it is in this gel form. When they add green light, it becomes a liquid. So what they did was they embedded a small molecule that can form sort of hydrogen bond and van der Waals interactions, and it can be kept locked in this transform matrix like here. But then imagine if you want this drug to be released, you simply add the green light, which will make it go into the cis form, and then you could release that small molecule. So I'm just showing you some different applications of using this tetrafluorinated azobenzene. Now, we're interested in using it for our application because we want to control oligonucleotide function, but there really wasn't a good way to synthesize it using like DMT phosphoramidite chemistry. So we had to think a little bit in terms of how to do this. And it wasn't terribly difficult. It would just required a few steps, right? So we started with like a difluoroaniline, and then we brominated with NDS. And then we did a cyanide displacement to install this carbon here, which we could then saponify and oxidize to this monomer here. And then we did the sort of classic carbodiamide coupling with this TBS protected linker. So that worked surprisingly, not surprisingly, worked really well. And then some nice methodology was published not too long ago by Lin and coworkers, where they could take these anilines and they could condense into azobenzene using like DBU and like NCS at like -78, really short period of time, like 20 minutes. And actually, just after some optimization, it actually worked really quite well. So we developed this symmetrical azobenzene, and then we could remove the TBS groups with acid. And then now we have our dial where we could add the DMT group and then the phosphoramidite. And I mean, this took some time, not because this didn't work well, but we were trying to initially add sort of different carbons here, not so much an oxidized carbon, but some of those synthetic methods were a bit challenging. But nevertheless, we're happy with this. And then we made our amidite, and then we could add it with an oligonucleotide. So now our model is, can we take the structure here, add green light, can we inactivate it, and can it stay inactive for long periods of time? That's the real goal. We don't want to have to keep green light on it for a long time. All right, so I'll walk you through the data. So we did a 48-hour experiment, and then when we added the sRNA to the cells, we added green light immediately for like 30 minutes. And then we turn off the green light for the rest of the 48 hours. And then when we measured activity, you can see that we get no activity. It's not working well at all. So that's suggesting that it's inactive, which is great. Now, what the next experiment is, we inactivated like before, but we added blue light a number of hours later to try to restore activity. And that's exactly what we see. We can see good restoration of activity comparable to our dark control. So we did that for 2 days. And then we also did the same experiment for 72 hours. Again, no activity. And then when we add the blue light, we can see activity. And again, with our dark control, we can see activity as well. So we're able to turn on the sRNA after it's been inactive. And then finally, similar to that first type of reversible experiment that I've shown you earlier, we want to see whether we could reversibly control the activity of the sRNA. So I'll walk you through this data. Number of different experiments here. So the green, I've shown you already, we can keep that inactive with a single green light injection. The next one is that green-blue, where we have blue light a few hours later, and we get activity. The next one is green and then blue at 24 hours. So we kept it inactive for 24 hours. And then 24 hours later, we added blue light for 15 minutes and then just kept it in the dark. So you expect it to be inactive for about a day, but then it has a day to restore some activity. So we would expect to see some activity, and that's exactly what we see with the purple one. And then the next one is what we call 1.5 times green, blue, green. So this is where we added green lights. A few hours later, we had blue light to activate it. And then we re-inactivated it again for the remainder of the experiment. So we wanted it to go through a few hours of activity, but for most of the time, it was inactive. And that's exactly what we see with these light green bars here. Then finally, similar to our previous experiment, we want to go through 2 full cycles, green, blue, and we would expect that the RNA should be active, but maybe not as much as the dark control, because at the end, the blue light would dictate its overall activity at the end. And that's certainly what we see. We see not strong gene silencing, but we certainly see a good dose-dependent gene silencing. And then our dark control, of course, is going to have the most pronounced gene silencing because it never went through green light inactivation, reactivation. So we're very excited about this result. And we did this experiment so we can reversibly control it. We also did this experiment for 72 hours. So I won't go through all the sort of those little details again, but we saw the same pattern. So inactive, active with blue light. When we add blue light 24 hours later, we get some activity. When it's through 1.5 cycles, inactive, and then 2 cycles, we get some activity, and then dark, we get more activity. So certainly there appears to be some good reversible activity with this. So based on this, we think we've developed some technology to control oligonucleotide function. And one thing that we think might be important as well is, whenever we're thinking of medicinal chemistry or drug development, we always want to think of like the dose, right? It's not just a half-life, but dose. So this could also give us some ability to control how strong we want the RNA to be in the body, because sometimes maybe different patients or different people might respond differently. So if someone responds too strongly to it, you could imagine maybe inactivating it for a little bit and then reactivating it. So this is maybe something sort of that could be used in the future. So just to conclude my presentation, we've gone from the system where we have azobenzene, UV light, inactivated visible light. We could do this reversibly, but we were, we were restricted by the short half-life and the fact that we use UV light. So we moved to this red light system where we could better control it, but we redshifted that pi to pi star transition. However, it had low thermal stability, very short half-life. So we needed better spatial control. So we then went to this tetrafluorinated group, which has high thermal stability. And then we can control this with green and blue lights for up to 72 hours using our system. So what we want to do is, so I won't kind of read this to you because I basically said these things. But what we're working on now through some collaborators is we're trying to examine the mechanism of action. And I just work with cell culture in my lab, but I have some colleagues who want to look at the effect in vivo. So we're looking at these Japanese Medaka fish, and these are fluorescently transgenic. So we want to look at how our RNAs can reduce in an in vivo model. They're also translucent, so we can control it with activity. So I don't have any data on this to present today, but in the future, hopefully we can move the system to higher order organisms. Yeah, so with that, I just want to sort of thank my group, of course, funding agencies. This is Matthew Hamill. He's a former PhD student who did most of this work with the azobenzene. And then certainly I have other students who are working on some of those other projects as well. And lastly, I'd like to just really thank you for your wonderful attention, especially to Professor Ramon Rodriguez for hosting me today. And I'll be happy to take any questions. Thank you very much. In English, please.
Did you promote any observations
Yeah, great question. I'll just answer here so people on the video can hear it. So the question was, when we add this azobenzene, do we remove the bases? And if so, how does that affect the hybridization, melting temperature? Yeah, so when we add the azobenzene, we remove two bases. And so naturally, without the complement, we're removing hydrogen bonds. So it does destabilize it thermodynamically. So typically for these RNAs with unmodified, we get melting temperatures around 70 degrees. So quite strong. When we add these azobenzene derivatives, we destabilize by about 15 degrees. So it's about 55 degrees. So it's a big drop, but it's still strong enough to remain a duplex once it's in the cell, because cells are 37 degrees. Yeah, but yeah, I didn't have time to talk about so much of thermodynamics. And then we also looked at circular dichroism to look at the secondary structure. And even though we've removed these hydrogen bonds, the structure remains intact, A-form RNA with them.
So we can write this and basic or
yeah. Yeah, so good question. So in our initial study with those simple linkers, if you recall those, yeah, one of them was very long. That replaced 3 bases. But if we place those linkers at different parts of the RNAs, they don't function very well. It only happens to be that middle region. So we think there's some what we might call like thermodynamic tolerance, where both ends are quite stable, but if you move it towards one end or the other, it gets too unstable and then it just doesn't function. So for the azobenzene, we just kept it right in the middle. You're welcome. Yeah, we thought about it. Yeah, like maybe on like electron donating groups and electron. Yeah. No, I think that's something we want to look at next. Yeah, for definitely those push-pull systems, and we could potentially get different control. Absolutely. So I would say probably the biggest challenge for us, is probably common for a lot of labs, it was the chemistry. That was the sort of the bottleneck, finding methods and being able to scale that up so we can make it. But I think moving forward, yeah, I'd like to try some of those push-pull azobenzenes for sure, now that we know the system works. Yeah, thank you for having me.